So all semester we've been talking about, the central dogma of biology, which pretty much states that DNA is going to serve as a template for RNA, and then RNA is going to serve as a template to make proteins. And so we're always going to start off with DNA. DNA is always going to be transcribed in a process called transcription, and you can abbreviate transcription Txp. Anyways, DNA is going to be transcribed into RNA, and then RNA is going to be translated into protein. And so it's going to be translated in a process called translation, which can be abbreviated TLN. So we're talking about transcription and translation here. They're two independent processes, but they go hand in hand with one another. And the important thing to remember is because we're starting off with DNA as a template, if you have an error in your DNA, that means you're going to have an error in your RNA molecule. And if you have an error in your RNA molecule, that means you're going to have an error in your protein. And if you have an error in your protein, that could lead to disease or something really bad happening, because mutations are mostly bad. 99% of the time mutations are bad. So if you have a mutation in your DNA, that's not only going to affect your RNA, but it's going to affect your protein too, because your protein is directly, or I guess I should say indirectly, dependent on what this DNA is. And so the whole process is known as gene expression. So if we're expressing a gene, we say we're turning on a gene. If we turn on a gene or express a gene, that means it's going to go through transcription, and then it's going to go through translation. So this whole process is termed gene expression or protein synthesis. There's multiple different phrases that describe this process, but these are kind of two of the big ones. So if you're talking about gene expression or protein synthesis, that's what we're talking about in this chapter. And so we're going to do a little bit of intro at the beginning, and then we'll focus more on transcription, and then when we come back on Tuesday, it'll be more of the translation. We'll wrap that up, the translation. Okay, so there's a little bit of difference between what happens in a eukaryotic cell and a prokaryotic cell. So remember, prokaryotes, they obviously don't have a nucleus, so that means transcription is not going to occur in the nucleus like it does in eukaryotic cells. So if we have our eukaryotic cell, there's our nucleus, there's all of our DNA, it only makes sense that transcription would occur in the nucleus because that's where the DNA is, right? So transcription in eukaryotes is always going to be in the nucleus. But since prokaryotes don't have a nucleus, transcription is just occurring in their cytoplasm or in their cytosol. Another thing that's going to be a little different is that eukaryotes, we're going to undergo a process called RNA processing. And it's basically what happens after transcription. We get this kind of rough, unpolished version of DNA. So in this RNA processing that we'll eventually talk about, we're going to add a couple things and kind of spruce up the RNA a little bit. Prokaryotic cells do not use this RNA processing, but eukaryotic cells will. And so then, after we have transcription and we have our RNA processing in eukaryotes, then that will turn into translation, which takes that RNA transcript that's been modified and now uses it as basically a code to build proteins. And so proteins are made up of amino acids, right? Well, each amino acid is determined by 3 nucleotides. 3 nucleotides at a time. So, as you can imagine, there's lots of different combinations of nucleotides, right? So you can have, if we're talking about RNA, you can have AUCG, you can have CCC, you can have GGG, you can have all sorts of combinations. But each combination of 3 nucleotides codes for a particular amino acid. So you're going to have your strand of RNA, and you're going to have these 3 little nucleotide sections of DNA. And those 3 nucleotides sections, that's called a codon. And so these codons, and examples of codons, like we said, could be AUG, could be CCC, any 3 combination of letters that are found in RNA. So there's 64 different options. So there's 64 different codons. But anyways, each codon codes for a particular amino acid. So once we transcribe our DNA into RNA, then whatever that sequence is, we just look at those little codons one by one and plug in the appropriate amino acid until we end up getting our protein. So that's just kind of a overview of what's going to be happening here. Okay, so as we mentioned, a bacterial cell, they're not going to have a nucleus, right? So their transcription and their translation is going to occur in the cytoplasm. And again, they're not going to have any RNA processing. Whereas in eukaryotic cells, we're going to start with our DNA, we're going to undergo transcription, we're going to make what's called a pre-mRNA. This pre-mRNA is what exists immediately after transcription, but before this RNA processing. So immediately after transcription, you're left with pre-mRNA. This is the unfinished version. And then we're going to go through RNA processing. We're going to add a few modifications. And after the RNA processing, you're going to end up with messenger RNA. And it's this messenger RNA or mRNA that's going to be used to build your amino acid sequence or to build your protein. So there's a difference between the pre-mRNA and the mRNA. The pre-mRNA is what you get immediately after transcription, and it's the unprocessed version of the RNA. And then messenger RNA, that's the modified, tweaked, clean, fun, friendly version of the mRNA. And that's what's going to be used on ribosomes in order to make these proteins. All right, so here's again kind of an overview of what we're talking about here. You have to remember that our genes in eukaryotic cells, our chromosomes are linear. So just long pieces of DNA, and along those pieces of DNA, we've got different genes, right? Gene 1, gene 2, gene 3, so forth and so on. And so along the pieces of the DNA, if here's my double-stranded DNA, what's going to happen is I'm going to use a template strand. And the template strand is always going to be the 3' to 5' strand. And so if I use the template strand, all I have to do when I transcribe my DNA, the theory behind it, you just put in its opposing nucleotide, its complementary nucleotide. But since we're making RNA and not DNA, all we're doing is we're switching the U's and the T's. So in this case, A would normally bind with T, right? But since we're making RNA, we replace that T with a U. C normally binds with G. C with G, A with T, but again, we're using uracil, uracil, uracil. So it's still based on complementarity. It's almost exactly like replicating DNA, except we're making a molecule of RNA instead of a molecule of DNA. So after you finish with transcription, you're going to get this mRNA, this pre-mRNA, and then it's going to go through processing. And afterwards, it's going to become messenger RNA. And then that messenger RNA is going to enter into translation. And what's going to happen, these ribosomes, which are responsible for making proteins, the way they work is they look at this codon. I told you codon is just a 3 nucleotide segment of your RNA. So they're going to look at this first codon. And this is kind of like, think of this as like Morse code or something. There's a little chart that y'all will have on your exam and that we'll talk about later on, but that gives you all the different codons and all the different amino acids that they code for. So this particular codon, UGG, that is always going to code for tryptophan. So the ribosome will read this first codon, bring in the amino acid tryptophan, and then move on to the next codon. The next one's UUU. This always codes for phenylalanine. So it's going to bring in that second amino acid, form a peptide bond, between here, and then look at the next codon. So the process of translation is just looking at this mRNA and reading it 3 nucleotides at a time. And each of those 3 nucleotides, or each of those codons, will code for a particular amino acid. So it's just like a secret code. This is always going to code for one amino acid. That's always going to code for one amino acid. That's always going to code for one amino acid, so forth and so on. So that's kind of the overview of gene expression. You transcribe your DNA to make RNA, and RNA is then processed to form mRNA, and then the ribosomes will read the mRNA 1 codon at a time, and then bring in the appropriate amino acid that that codon codes for. And so if we were going to make or transcribe a piece of DNA, I just kind of made this up, but ATG, CGT, AAG, so if we were going to transcribe that, that's the template strand, again, you would just put in the complementary nucleotide. And just remember not to put in the T's. So there will be a question like that on the exam. It's a multiple choice question. It says, if this is your piece of template DNA, what does the RNA look like? So it's literally just putting in the complementary nucleotide, but making sure there's no T's there. So y'all think y'all can handle that? Okay. You want to do an example, or are we good?
So is that standard? It's always...
Yeah, as long as you're using the template strand. So you're always going to be using the 3' to 5' strand as the template. Other questions? Okay, so we've already kind of talked about what a codon is. It's this set of 3 little nucleotides. And there's going to be 64 different codons. 61 of them are going to code for a particular amino acid. The other 3, these are going to be a special type of codon called a stop codon. Now, a stop codon, it's still a codon, but it doesn't correspond to any particular amino acid. Instead, what happens is, so imagine we've already gone through transcription. So we have our RNA molecule right here, our messenger RNA. So we've got all these, codons, which will bring in certain amino acids. So we're making our protein chain, so forth and so on. And then eventually, the mRNA is going to end with one of these 3 stop codons. And by the way, you are responsible for knowing all your stop codons. They are UAG, UGA, and UAA. So whenever you see any one of those 3 at the end of an RNA molecule, that tells you that translation is over. So it's literally like a big stop sign. It's like, okay, this is the end of the gene. You can go ahead and stop making protein now. So that's all it is. It tells the machinery of translation that you're done making protein. Just like we have a stop codon, we also have a start codon. So a start codon is where you're going to start making protein. And so every single gene, every single mRNA transcript in your body is always going to start with the same start codon. There's only one of them, and it's AUG. AUG is included in one of these 61 that codes for a particular amino acid. So AUG is always going to code for a methionine. So if every single piece of mRNA in your body starts with AUG, what does every single protein in your body start with? Methionine, right? Because AUG only codes for methionine. So every single protein in your body starts with the same amino acid. It always starts with methionine. Because the start codon, there's only one of them, and all the start codons are the same, which means you're always going to start with methionine. So what that means is that every piece of DNA, if you're talking about the template strand, so the 3' to 5' strand, if the RNA is going to start with AUG, what do you think those first 3 nucleotides are going to be before you transcribe it into AUG? So we went from DNA to RNA, now go backwards from RNA to DNA. TAC, right? So every single gene in your body, every single beginning of a gene in your body starts with TAC. And so when you transcribe that, you get AUG. And then when you translate that, you're going to end up with methionine as your amino acid. And so that's the only basic truth to translation. You're always going to start at a start codon, and you're always going to finish at a stop codon. And you're responsible for knowing your start and your stop codons. So these are the stops, and there's your start. So you got to memorize 4 of these codons. All the rest of them, I'll have a chart for you. Yeah?
For the stop codon, we only need one of them, or we only
Yeah.
need 3.
You only need one of them. There's just 3 different options. But there's only one start codon. And so when we're reading this codon, in order to make our protein, it depends on how we read it. So if I have a piece of mRNA that's UAC, GAA, UCG, AUC, the way we read the codon, this will be one codon, this will be the second one, this will be another one, so forth and so on. But if we insert an extra nucleotide or change the way we read the piece of RNA, so here, instead of reading this as an entire codon, we've shifted it 1 nucleotide to the left. So now this becomes a codon. It's still the same sequence, so it's still UAC, it's just been shifted. So the way you read a codon determines which amino acid you're going to get. And so when we're doing things normally, we say that we're reading the codons using the correct reading frame. So the reading frame is when you get the correct codon that codes for the correct amino acid. This is usually what happens. But sometimes you can get some sort of insertion or some sort of deletion of nucleotides, and that's going to mess up your reading frame because it's going to mess up which amino acid is supposed to be in your protein. So you can see that these 3 different reading frames are all going to produce different proteins because they all have different codons, right? So each protein is going to be different depending on how those codons are read. So in this case, this is one codon, this is one codon. And then we change the reading frame here by shifting everything. So instead of UAC, we still have UAC, it's just the U's been shifted to a different codon. So now this becomes our codon. And then that changes that codon, and then that changes that codon. So everything gets shifted over one way or the other. So that's what we say is causing a change in the reading frame. So don't get too caught up on this. I see confused look on your faces. This will come into play when we start talking about mutations. There are reading frame or frame shift mutations where you have an insertion. So if I inserted an extra nucleotide right here, let's just say I inserted an extra U, then what would my first codon be? It'd be UUA, right? So if I inserted an extra U right there, it would be UUA. Then that means this C would have to go over here and be a part of that codon, so CGA. Then that means this guy would have to pair up with those guys. So it's going to change the way you're reading your nucleotides. If you change the way you're reading your RNA, that's going to change the proteins or the amino acids that are going to make up that protein. Does that kind of make sense? Okay. All right, so here's this codon chart that I speak of. And so on the exam, like I said, this will be provided for you, but this will be whited out, and all the stop codons will be whited out. But if you're smart enough, you should be able to figure out what they are anyways, because it'll still start with an A, it'll have a U as the second one, and then it'll have a G. So the way you read these, this is the first nucleotide. So all of these are going to start with U, all of these are going to start with C, this is the second nucleotide, so the second nucleotide always going to be U, C, so forth and so on. And then finally, the last nucleotide is here. So if I want to look at phenylalanine, U. So I'm going to start with U, okay? I know that U is the second one. And then phenylalanine, you have two different codons that code for phenylalanine. So phenylalanine could be UUC or UUU. And so you might notice that there's not one codon for one amino acid. There's multiple codons that code for the same amino acid. So in other words, if I'm a ribosome and I'm going down my mRNA and I'm making protein, I could come across either one of these, any of the four, and I'm still going to put in the same amino acid, so threonine. So that means that the genetic code, there's a lot of redundancy, right? There's a lot of excess. There's extra ways, there's multiple ways of making one protein. You can make it with different codons, but it's still going to be made up of the same amino acids. And again, these stop codons, they don't code for a particular amino acid, which is why they just say stop here. So once, translation is moving, the ribosome's going, it's making protein, and it runs into one of these three guys, and it basically shuts off the system and stops making protein. It tells you basically, all right, this is the end of the gene, you don't need to make any more protein anymore. All right, so we're going to start focusing on transcription at this point. And so transcription, again, is when we're starting with a template of DNA, and we're going to convert it into a piece